Good morning, sir. My name is Rosalinda Ballesteros, and I am going to discuss mutation and gene regulation. First, mutation is an alteration in the nucleotide sequence of the DNA of the genome of an organism, virus, or extra chromosomal DNA. Viral genomes can be of either DNA or RNA. Mutation can be inherited. Parents that have mutation can pass them to their offspring. Germline mutations are present in every cell of an individual, including the egg sperm used in the production of offspring. Next, de novo new mutations occurs by chance in one or a few eggs or sperm or just after fertilization and are not present in every cell of a parent. Next, acquired. Environmental agents called mutagens can alter DNA. Types of mutation. First, point mutation, a change in a single nucleotide. Second, chromosomal mutation. Insertion occurs when a portion of one chromosome has been deleted from its normal place and inserted into another chromosome. Deletion changes the number of DNA bases by removing a piece of DNA. Next, insertion mutation. One or more bases are inserted into the original DNA based on sequence. Duplication consists of piece of DNA that is abnormally copied one or more times. Frameshift mutation caused by insertion or deletion for a number of nucleotides, changing the reading frame, changing the reading frame, and resulting in a completely different translation from the origin. Inversion. Inversion is a type of mutation where a sequence of nucleotides in the DNA is reversed. Next, gene regulation is the process of controlling which genes in a cell's DNA are expressed, used to make a functional product such as a protein. Different cells in a multicellular organism may express very different sets of genes, even though they contain the same DNA. They are the various forms of gene regulation. Mechanism for controlling which genes get expressed and at what levels. However, a lot of gene regulation occurs at the level of transcription. Anatomy of an operon. Operons aren't just made up of the codon sequence of genes. Instead, they also contain regulatory DNA sequence that control transcription of the operon. Typically, these sequences are binding sites of regulatory proteins which control how much the operon is transcribed. The promoter or site where RNA polymerase binds in one example of regulatory DNA sequence. These are the liver cell and neuron. Yan. Mayroong left panel. Left panel, liver. The liver cell contains alcohol, dehydrogenases, proteins. If we look in the nucleus, we see that an alcohol dehydrogenase gene is expressed to make RNA, while neurotransmitter gene is not. The RNA is processed and translated, which is the alcohol, dehydrogenase, proteins, and are found in the cell. Yung right panel, neuron, the neuron contains neurotransmitter proteins. If we look in the nucleus, we see that the alcohol hydrogenase gene is not expressed to make RNA, while the neurotransmitter gene is the RNA is processed and translated, which is why the neurotransmitter proteins are found in the cell. And that's all. Thank you for watching.